For most of the 20th century, there was a quiet certainty among population geneticists. 2,000 years of diaspora, they assumed, should have erased any meaningful genetic thread connecting Jewish communities scattered across 5 continents. Centuries of exile, migration, conversion, and intermarriage had surely dissolved whatever biological signal once existed. The religion survived. The genes, surely, did not. Then the DNA results came back. And they made no sense. Because what geneticists found was not erosion. It was preservation, on a scale that defied everything they thought they understood about how populations change over time. A genetic signature so distinctive that a computer algorithm could separate Jewish individuals from non-Jewish Europeans with 100% accuracy, without a single error, across hundreds of random samples. That finding alone was extraordinary. But it was only the beginning of a far stranger story, one that would lead from ancient priests to medieval bones, from a synagogue observation to a remote African tribe, and, ultimately, to a number that would rewrite how we understand survival itself. That number is 350. To understand why that number matters, you first have to understand what geneticists thought they already knew. By the 1980s, early studies using blood group markers and serum proteins had established a rough picture. Jewish populations, whether from Morocco, Iraq, or Poland, seemed genetically closer to each other than to their non-Jewish neighbors. The signal was there, but it was faint, like trying to read a watermark through layers of paint. The tools simply could not resolve how much of the similarity was ancient shared ancestry, and how much was coincidence, or sampling error, or the normal genetic background of the broader Middle East. And there was a deeper assumption beneath those early studies, one that felt so obvious that nobody questioned it. Judaism, most scholars insisted, is primarily a religious and cultural identity. People convert. Communities absorb outsiders. Over 2 millennia, the reasoning went. Even if there was an original Levantine population, intermarriage with Europeans, North Africans, and Central Asians should have diluted any founding genetic signal beyond recognition. The math seemed simple. The conclusion, settled. It was wrong. What nobody could see, with the tools available at the time, was that the picture was incomplete for reasons that had nothing to do with Jewish history and everything to do with the limits of the science itself. Consider what researchers were actually working with. Blood group markers analyze a handful of genetic variants out of the 3 billion base pairs in the human genome. It is like trying to reconstruct a city from 3 photographs taken through a keyhole. You get impressions, you get hints, you do not get a map. Y chromosome and mitochondrial DNA studies, which emerged in the 1990s, were sharper, but they still represented tiny slivers of the full picture. 1 paternal line and 1 maternal line out of thousands of ancestral pathways. Useful, certainly, but profoundly narrow. And those narrow windows told contradictory stories. Y chromosome data showed strong Middle Eastern paternal ancestry among most Jewish groups. Mitochondrial data, tracking the maternal line, was far more ambiguous. Some lineages pointed to Europe, others to the Near East. The father's story and the mother's story did not match. Without the ability to read the whole genome at once, there was no way to reconcile them. Researchers were forced to choose sides, and for years, the field fractured into competing camps, each armed with partial evidence and absolute conviction. 3 things kept the real answer hidden. First, the tools could not read enough of the genome. Second, no one had ancient DNA from Jewish individuals to compare against living populations. And third, an assumption had calcified into fact, the belief that 2,000 years was simply too long for any genetic signal to survive in a minority population surrounded by a genetic ocean of host communities. The technology that would shatter all 3 barriers arrived in the late 2000s. High-density SNP arrays, capable of simultaneously reading over 500,000 genetic markers per person, changed everything. And the story that emerged from those arrays was unlike anything either camp had predicted. It began, oddly enough, not in a laboratory, but in a synagogue in Toronto. Karl Skorecki was a nephrologist, a kidney specialist, and a Kohen, a member of the Jewish priestly caste that traces its lineage patrilineally to Aaron, the brother of Moses. One morning in the early 1990s, he watched a Sephardi Jew called to the Torah for the priestly blessing. The two men looked nothing alike. Skorecki, pale-skinned and Ashkenazi. The other man, darker, Sephardic, of North African descent. Yet both carried the same oral tradition, the same family claim passed father to son for over 100 generations. Skorecki, staring at his own hands, asked a question that should have been trivial. Could that claim possibly be tested? He contacted Michael Hammer at the University of Arizona, a specialist in Y chromosome research. Together, they collected cheek swabs from 188 Jewish men, Ashkenazi and Sephardi, priests and non-priests, from 3 continents. They extracted the Y chromosome DNA, a fragment of code passed exclusively from father to son, and compared the results. The data locked into a pattern that should not have been there. Self-identified priests, men named Cohen, or its variants, from communities separated by 1,000 years and thousands of miles, carried a shared genetic signature at a frequency that dwarfed the general Jewish population. Skorecki and Hammer called it the Cohen modal haplotype. It appeared in roughly half of all Kohanim tested. The odds of this happening by chance were less than 1 in 10,000. Before we see where this evidence leads next, if you are finding this story as fascinating as we are, subscribe so you do not miss the discoveries still unfolding. When a follow-up study calculated the divergence time of the Cohen modal haplotype, the estimate landed at approximately 3,200 years, give or take a millennium. That date range sits precisely over the era described in Exodus, when Aaron was anointed as the first high priest. 100 generations of oral tradition, it appeared, had left a detectable trace in living DNA. But the Cohen study was only the first tremor. The earthquake came in 2010. That year, 2 independent research teams published genome-wide analyses simultaneously. Doron Bihar, working with collaborators across 14 institutions, genotyped individuals from 14 Jewish diaspora communities and compared them to 69 non-Jewish populations using high-density arrays reading over 500,000 genetic markers per person. Meanwhile, Gil Atzman at Albert Einstein College of Medicine ran a parallel study on 7 Jewish groups: Iranian, Iraqi, Syrian, Italian, Turkish, Greek, and Ashkenazi. Both teams reached the same conclusion, independently. On a principal component plot, a graph where each dot represents a person's entire genome, Jewish communities from across the globe collapsed into a remarkably tight cluster. They did not scatter among their host populations. They did not blend into Europeans or Middle Easterners. They sat in a distinct space, overlapping with Druze and Cypriot samples, positioned between Europe and the Levant, but belonging wholly to neither. Ashkenazi, Sephardi, and Mizrahi Jews, separated by geography and centuries, clustered together as though the diaspora had barely touched their core genome. What the evidence showed was unambiguous. Genome-wide data confirmed shared Middle Eastern ancestry across virtually all major Jewish groups, with variable but measurable admixture from host populations. What it ruled out was the idea that Jewish identity was a purely cultural phenomenon with no biological coherence. The genetic clustering was too tight, too consistent, too reproducible across independent datasets and different analytical methods to be an artifact. What it implied was that endogamy, the cultural practice of marrying within the community, had functioned as a biological seal for roughly 2 millennia, preserving an ancestral genetic signature through dozens of generations of exile. And then came the number. In 2014, Shai Karmi, then at Columbia University, led a consortium that deep-sequenced 128 Ashkenazi Jewish genomes. By analyzing long stretches of identical DNA shared between ostensibly unrelated individuals, stretches that could only exist if those individuals descended from the same small founding group, the team calculated the effective size of the Ashkenazi bottleneck. 350 people. The entire modern Ashkenazi Jewish population, over 10 million individuals on every inhabited continent, can trace its ancestry to a founding group of roughly 330 to 350 people who lived in Central Europe approximately 700 years ago. The expansion that followed was explosive, a growth rate of 16 to 53% per generation, producing a vast population from an almost impossibly narrow genetic base. As Columbia's Itzik Pe'er put it, among Ashkenazi Jews, everyone is a 30th cousin. This bottleneck explains what had puzzled medical geneticists for decades: the elevated frequency of specific disease mutations, Tay-Sachs, Canavan disease, certain BRCA1 breast cancer variants, that run through Ashkenazi communities at rates far above the general population. These were not random accumulations. They were the legacy of that tiny founding group, rare mutations carried by a few of those 350 people, amplified by rapid population growth in the centuries that followed. But when did the bottleneck actually happen? For years, researchers assumed it coincided with the Crusade-era pogroms or the Black Death massacres of the 1300s. Ancient DNA would soon challenge that assumption. In 2022, an international team of over 30 researchers published the largest ancient Jewish DNA study to date. They extracted DNA from teeth of 38 individuals buried in a medieval Jewish cemetery in Erfurt, Germany, dating to the 14th century. The results were startling. Medieval Erfurt Jews were more genetically diverse than modern Ashkenazi Jews. The bottleneck signature was already present in their genomes, meaning the population crunch occurred before the 1400s. Separately, analysis of 12th century remains from a well in Norwich, England, individuals almost certainly killed in a pogrom in 1190, showed disease mutation frequencies nearly identical to modern Ashkenazi populations. The bottleneck had happened before even those medieval victims were born. The field has not settled on a final date. Some models place it as early as the 8th or 9th century, when a small group may have crossed the Alps into the Rhineland at the invitation of Charlemagne. Others point to a longer, slower constriction. But the genetic evidence is clear. The signature was already set by the 1200s. There is also sharp disagreement about the maternal side of the story, and this is where the debate still burns. In 2013, Martin Richards and his team dropped a provocation into the field. After analyzing mitochondrial DNA, they argued that roughly 80% of Ashkenazi maternal ancestry originated in Europe, not the Middle East. The implication was stark. Jewish men from the Levant married local European women, and those women's lineages dominate modern Ashkenazi mitochondrial DNA. If Richards was right, the story of Jewish genetic continuity was only half true, preserved on the father's side, but largely European on the mother's. Doron Bahar pushed back hard. He argued Richards's phylogenetic methods were flawed and that the deepest maternal roots were still Near Eastern. A 2014 study by Fernandez and colleagues added fuel, finding Near Eastern frequencies of mitochondrial haplogroup K that flatly contradicted a predominantly European maternal origin. The field remains divided. Meanwhile, the more controversial Karzar hypothesis, proposing that Ashkenazi Jews descended primarily from a converted Turkic empire in the Caucasus, has found almost no genomic support. Multiple independent analyses have placed Ashkenazi ancestry firmly between the Middle East and Southern Europe, not Central Asia. What is not in dispute is the overall architecture. Jewish populations worldwide share a detectable Middle Eastern genetic core. The amount of admixture from host populations varies, highest in Ethiopian and Indian Jewish communities, lower in Ashkenazi and Sephardi groups. But the core persists. And then there is the part of this story that nobody predicted. Deep in southern Africa, scattered across Zimbabwe and South Africa, lives a Bantu-speaking people called the Lemba. Numbering around 70,000, they practice male circumcision, avoid pork, observe endogamy, and bury their dead facing north, toward Jerusalem. For generations, they have claimed descent from Jews who sailed from a place called Senna, possibly Sana'a in Yemen. Most academics dismissed this as folklore, until the Y chromosomes were tested. In 2000, Mark Thomas and Tudor Parfit analyzed 399 Y chromosomes from Lemba men alongside samples from Bantu, Yemeni, Sephardi, and Ashkenazi populations. The Lemba Y chromosomes split cleanly into two lineages, one Bantu, one Semitic. And in the priestly Buba clan, the family that oral tradition says led the Lemba out of Israel, 50% of men carried the Cohen modal haplotype. That is a higher frequency than in the general Jewish population worldwide. Today, those 350 Ashkenazi founders are not just a statistic. They are a challenge to how we think about cultural survival. Somewhere in medieval Europe, a community smaller than the population of a single apartment building carried within its genome the mutations that would affect millions of descendants, the religious traditions that would outlast empires, and a genetic signature so distinct that a machine can read it 7 centuries later, with perfect accuracy. If this chapter of hidden history has fascinated you, subscribe to explore more stories the evidence is only now revealing. The bones from Erfurt still sit in a lab in Jerusalem. The Lemba still bury their dead facing north. And somewhere in the genomes of 10 million living people is a record written not in ink, but in nucleotides. A record of 350 strangers who built a civilization from almost nothing. If an oral tradition survived 100 generations in DNA, what else might be encoded in genomes we have not yet thought to test?